Alright, we're going to do an afternoon journal club today, ladies and gentlemen. An afternoon journal club today. We're going to be covering a relatively recent paper, so it won't be necessarily that great for beginner neuroscientists, but you could get a lot out of it. Am I in your way, sir? Sorry about that. No, I wasn't. I was leaving. That guy looks like he's working hard. So, the paper today is a 2019 paper by Bernardo Rudy's lab. And Bernardo Rudy is, well, he's sort of a elder statesman among neuroscientists today. Bernardo Rudy is a... You too? Yeah, yep. He's a man among boys. He's a giant, right? He's a beast. And his laboratory does beastly stuff. And very specifically, his laboratory does very thorough beastly stuff. This paper is an example of thorough discovery in neuroscience. That's why I'm covering this paper today. It's thorough discovery. He doesn't leave any doubts left over. He's showing you a result, and the result is going to be very hard for you to refute. And in fact, the results as they stand will require you to change your model of the cortex, essentially, to fit the model that incorporates 4 different interneuron subtypes in layer 1. 4 unique interneuron subpopulations are found in layer 1 in this paper. And so over the course of the 9 or so figures that this paper has, you're going to be thoroughly convinced that in layer 1 there exist 4 functionally distinct populations. And I think that's a pretty significant finding given the fact that we really didn't know very much about layer 1 interneurons before this paper. We knew that layer 1 interneurons were there. We knew that they might be special, but layer 1 was always sort of enigmatic because when you look at slices of the cortex, layer 1 doesn't appear to have very many cells in it. And I think for a long time in cortical research, the cells of layer 1 were just thought to maybe not even belong there. They were just, maybe they weren't even, they were just didn't migrate completely or were supposed to be somewhere else. So this paper now is going to clearly define 4 functional subtypes within layer 1. Holy crap, she was turning in front of me. She only looked at the last minute. So 4 unique interneuron subpopulations in layer 1. The first figure is just going to make the argument that the mouse that they use has an enriched signal in layer 1, so that they could use this mouse to target layer 1 cells, and that it makes sense to use this mouse. It's an NPY linked to GFP, and so they can label all cells in layer 1 with this NPY mouse, but it's not all the cells in layer 1. And so in this first figure then they break down the fact that there are 2 types of cells basically in layer 1, NPY positive and NPY negative cells in this mouse. And thank you for riding in the frickin' bike lane, you dipshit. In this mouse there are 2 basic cell types that get identified, NPY positive and NPY negative cells. And then amongst those cells there's a certain subpopulation that is late spiking, and there's another, and those two populations, NPY positive and NPY negative, have a subset of cells which are late spiking. And so then you can already imagine a scenario where there's 4 groups of cells in layer 1 in this case, right? Can't tell. Is there somebody behind me all of a sudden? What is that noise? Sounded like there was someone behind me for a minute. Okay, so they're going to use this mouse. And then in figure 2, they're going to go a little bit farther and they're going to separate these cells by marker and show that there are 4 useful markers to separate the cells which they first have only been able to separate as NPY positive, NPY negative, and then some spiking deal. And the markers they're going to use are NDNF, and NPY, nicotinic receptors, and VIP. And so now you're hearing some, for those of you in the game, you're hearing some more familiar markers here. No. Seriously? Seriously? Okay, so thought that would go. Oh, I see. Can't go. So now they're going to break this down into 4. And these 4 molecular categories sort out in the staining. So when they stain, the stains largely do not overlap in layer 1. And that's what they're showing you here with this immunohistochemistry figure, is that the staining in layer 1 tends to not overlap, so you get non-overlapping cell populations. And again, this sort of indirectly confirms the existence of 4 populations. And the 4 populations tend to segregate on, again, NP, what is it called? NDNF, I think it is. Gosh, it's hard for me to remember. NDNF, NPY, nicotinic receptors, and basal intestinal protein. So basal intestinal protein was already a marker for layer 1 interneurons. So this is not surprising. It's nice that this is incorporated in there. And then NDNF, NPY, and nicotinic receptors. So then in figure 4, well, let's stay with figure 3 first, because figure 3 is also going to break down the spiking patterns and show that the spiking patterns of these neurons are all different, and that there are certain aspects of each of these spiking patterns that make them unique. And now, using physiology like this to classify neurons is not new. Using electrophysiology is actually one of the gold standard methods. And now here, Rudy's lab is taking the painstaking effort to Okay, so I guess we're going to record now again. I hope this works. Call from my doctor. So figure 1 shows the mouse they're using and the fact that they can more readily target layer 1 cells in that mouse. They're mostly NPY positive and NPY negative neurons with some different in their spiking pattern. Second figure suggests that there's probably some molecular markers that line up with these 2 to 4 groups of neurons, including the molecular markers of NDNF, NPY, nicotinic receptors, and the basal intestinal protein VIP, a previous marker of layer 1 interneurons. Now we go on to figure 3, where we show the electrophysiological groups that correlate with these markers. And so here you see very unique spiking patterns with very specific attributes of the physiology correlating with the 4 groups. I won't break that down, you can find that in the figure, but the low threshold, or the late spiking phenotype, for example, correlates with only one of the 4 groups, And this makes for a very interesting finding now, because if the four groups are just four groups, but they aren't physically or physiologically distinct, then the four molecular groups might really not mean very much. But the fact of the matter is that they are very electrophysiologically distinct. And these markers are relevant markers, of course. Nicotinic receptors or no nicotinic receptors means you respond to cholinergic inputs and not. So the idea that these two are segregated, that's a significant segregation. Activation by choline and not by acetylcholine is a significant functional segregation. And so here, again, they nail it with this figure. And so then they are already, in my mind, presenting a beastly figure. But now figure 4 goes on to show that the morphology of these subtypes in layer 1 is different. That their dendritic projections and their axonal projections are different. And these fundamental differences are obviously part of their physiological differences. But what you need to see here is that the correlation between the four groups and the four distinct profiles is so sharp. And that's what makes this finding so nice and so easy to reconcile with the current literature. Because there's nothing here that's contradictory, nothing here that's a little controversial. It's just four interneuron groups that you didn't know about before, and segregating on very relevant markers, which also makes it very easy to accept as fact, accept as canon. So after they do the morphologies in figure 4, then in figure 5, they show all the morphologies of all the subtypes. And so you can see some really interesting patterns there that maybe it's not immediately obvious from the morphological examples given in figure 4, where they just have one and they show you a graphical representation of the distribution of the axons and dendrites. In figure 5, your own brain can start to find, for example, the axonal branches in 5A, or the lack of axonal projections to deep layers. And you can see it across the groups. And I want to emphasize here that filling the axon of interneurons is not trivial. Filling the axon of this many cells is not trivial. On your left, sir. Thank you, sir. You too. Did I say on your left? I guess I was on his right. So figure 6 and figure 7 delve a little bit more deeply into the morphological differences and the statistics associated with them. I'm not going to go into this in detail because I don't think it's possible to do it very well on the bike. But those figures provide further statistical argument for the fact that these are distinct groups, and distinct groups based on morphological differences on top of the markers and on top of the electrophysiological clustering. So after we go to 6 and 7, figure 8 is a beautiful example of how the cells might differentially contribute to the local microcircuit. So here they just look at 2 of the interneuron populations in layer 1, and they demonstrate that they have a differential connectivity with pyramidal cells in layer 2. And that differential connectivity results in almost 75% connectivity with one group and zero connectivity with another group. And so again, they've already shown you that these appear to be different interneurons. But now they're giving you even more evidence that functionally they're different. They sit in a different place in the microcircuit relative to other cells. And I think this is what makes this paper so cool, because I think these are the kinds of puzzle pieces that I'm interested in finding. How are specific types of interneurons connected to specific groups of pyramidal cells in the cortex? And are any of these patterns unique, and any of these patterns repeat through the cortex? So that we can start to understand how rhythm can emerge from the cortex, how rhythm can be used to, rhythm can be used to select cell groups, rhythm can be used to isolate cell groups, and to functionally elevate cell groups. So this kind of differential connectivity will be the basis for these kinds of microcircuit studies, and will be the basis for our computational understanding of how the cortex can work. Henry Markram can't make a relevant model of the cortex until he knows who's connected to who and what those synapses look like from a plasticity perspective, from short-term dynamic perspective, which we talked about in the last paper, and in the Reyes paper, which I still haven't put up. So then, of course, the last figure, figure 9, does the same thing that figure 8 did, but now it's going to look at two of the interneurons, pairs of interneurons in layer 1, and show that those pairs have a directional connectivity bias. And again, all we're trying to do here is show specificity. If these were all connected in the same way to all the same cells, then making an argument that they play different roles in the microcircuit would be more difficult. And the observations of having different molecular markers or different physiology would mean less without these paired data. These paired recordings demonstrate unequivocally that they are functionally different groups. And so this is what makes, in my mind, figures 8 and 9 are the checkmate figures. And so here you got a paper which expands the palette that the cortex works with, the basic erector set from which all cortical microcircuits are made. And it expands it in a way that, you know, finding a new subgroup in layer 5, I don't think would be as cool a finding, because we already knew that they were there. Layer 1 was oftentimes characterized as sort of an axon layer. Cortical connections, neuromodulators. You see that in the figure 1 in their cartoon. And so it's a good reason to look in layer 1. In reality, it took somebody like Bernardo Rudy to see that layer 1 was just wide open. Basically a very low-hanging fruit for anybody with the stamina to do it. And the way they did it was pretty cool. They found a mouse the NPY GFP mouse that was somewhat enriched for layer 1. And so they could patch green and non-green cells in layer 1 and already expect some segregation. And they saw it already with just that mouse and just those patchings. And then they broke it down further by molecular marker and correlated that with physiology, which seemed to directly verify 4 groups in layer 1. Then in a series of figures, they analyzed the morphological difference between these groups. 4, 5, 6, 7. These are like now a gold standard for single cell patch clamp morphology, electrophysiology analysis. I mean, nobody's doing it better than this. You can do it as well, but you can't do it better than Bernardo's lab. So I hope you find this paper helpful. I definitely think it's a paper that everybody in neuroscience needs to know of. You ever talk about cortex? Ever talk about layer 1? And ever hear anybody say that there's no cells in layer 1? Now you know. I can't move over because there's holes in the road there. Whenever somebody's coming up the hill on that one particular stretch, I'm always very close.